All right, this is part 2 of our photosynthesis lecture. I'm going to see if I can get this to upload to YouTube. I was having problems with the other video, so I'll give it a shot. Tomorrow on your quest, you're going to see a diagram of a chloroplast, and you are expected to know which reactions take place in which part of the chloroplast. Remember, the chloroplast is a double-layered organelle, and it has its own DNA, so it is capable of replicating itself or copying itself independently from the rest of the cell. So this is one of the factors that contributes to the theory of the endosymbiont, and that's a big word. Endosymbiont, so a symbiosis within, where the chloroplast was thought to be potentially its own, originally a prokaryotic cell on its own, which was then engulfed by another prokaryotic cell, and so then you got a, sorry, that's my phone, then you got a eukaryotic cell with, see, there's a little chloroplast there going in and being engulfed, and so then they developed a symbiotic relationship between the two cells. And so you had one symbiotic organism within another. What contributes to this is not only the fact that a chloroplast has its own DNA, but it's also a double-layered organelle. So you see here, if this organelle, this is the chloroplast here, a very poorly drawn chloroplast, if that chloroplast was engulfed by another prokaryotic cell, then it would have had its original cell membrane, and then it would have had a new cell membrane, and that's what makes it double-layered. So that's the idea. Mitochondrial cell organelles are also double-layered, and they do have their own DNA. So another organelle that is thought to have been once its own prokaryotic cell. So we have two chemical reactions. We have the light-dependent reactions and the Calvin cycle. Okay, the light-dependent reactions take place across those thylakoid membranes. And remember, the thylakoids are stacked, so the thylakoids are the individual little pancakes, and the stack of the pancake is called the granum. And grana, I believe, is plural, singular is granum. And then the Calvin cycle takes place in the stroma, which is the fluid outside. Okay, moving on here. So if I went to the store and bought a green colored light, and I placed my plant underneath that green light, and I closed it tightly, blocking off all other light, will the plant be able to do photosynthesis? I want you to think about that. Would the plant be able to do photosynthesis? And I want you to think back to the graph we had a couple of slides ago, which showed the reflective parts of the spectrum from chlorophyll A and B. And think about that before you answer the question. Think to yourself. Okay? So as we look at an overview of photosynthesis, we have, in the process of photosynthesis, the plant is converting light energy from the sun, into chemical bond energy. Okay, specifically chemical bond in covalent bonds. Okay, covalent bond energy. And if you remember, when we create a chemical bond, it requires energy, and then chemical energy is released when a chemical bond is broken. So, this is not responding here. Hold on a second, I'm having some technical difficulties. Here we go. Okay. So we have two reactions, light reactions and light independent reactions. We don't like the term dark reaction. Remember, we don't like the term dark reaction. It's also known as the Calvin cycle. Okay, and the general equation for photosynthesis, can you fill it in? Okay, and this looks awfully messy here, so let's get rid of this. We'll ignore, oh, what did I just do? No. Don't you love it when I have problems with my computer while I'm recording? Isn't that fun? let's ignore this here and let's put this over here. That's just a glitch. So CO2 plus H2O gives you C6H12O6 plus O2. But is this equation balanced? Can you balance it? We need to have 6 CO2, 6 H2Os, and 6 O2s over here. Now, in your textbook, it might show that you have water not only on the left side of the equation, but you also have water on the right side of the equation, because water is used and produced in the process of photosynthesis. But that's something that you can look back on for AP Bio. We don't need to worry about that here. 5 things that are needed for photosynthesis: water, sunlight, carbon dioxide, chlorophyll, and the other pigments. Enzymes, something like 30 different enzymes are needed for photosynthesis. It's crazy. 30 different enzymes and proteins. So a huge amount of biochemical reactions going on. Remember our lock and key model. Remember, enzymes can only do one thing. They can only metabolize one chemical reaction. So that's why we need to have 30 different ones, because they're all doing different things at different stages. All right, our light-dependent reactions are taking place in the thylakoid membranes, across the membrane. The electrons are being excited and going to a higher energy state from the splitting of water. Water gets split, oxygen comes out. Those electrons go into the chlorophyll molecule, they get excited, they come down, and the electron transport chains, and they give off a little bit of energy. It happens first in photosystem 2. Then those electrons get excited again to a higher energy level, come back down in another electron transport chain in photosystem 1. Why do we have 2 before 1? Well, the photosystem number 1 was discovered first, and then they discovered photosystem 2. But in reality, the electrons from photosystem 2 are what feed into photosystem 1. Without photosystem 2, the whole thing would break down. The end result is that you have a last electron acceptor here at the end of photosystem 1, and it's an NADP molecule, and it accepts the extra hydrogen, and it becomes NADPH. And that then feeds into the Calvin cycle. Okay, so we get ATP and NADPH, hydrogen's trapped, oxygen is released as a result. Okay, so here's a nice picture. Okay, electron transport chain, energy splits, and what this really isn't showing here is that it actually goes through photosystem 2 and then photosystem 1, and it's in photosystem 2 where the water is produced. But both the NADPH, see the little red star, and the ATP, both are used in the dark reactions. The electron transport chain, what is that? The electron transport chain is a way that, I love this little picture here. There's a great little video, hopefully the link is still working, it's not broken, but in the leaf, the sunlight energy basically just pours, and it just gets passed from one molecule to another, one biochemical molecule to another, and it goes from a higher energy state to a lower energy state. This is highly efficient, this process. And when we split that water molecule, that's where we get our free oxygen that we breathe. And then the hydrogen is trapped by that NADPH. And remember, we've got a chemical bond holding that hydrogen in place, and so when we break the hydrogen off of the NADPH, energy is going to be released later on in the chemical reaction. Just like when we pull off a phosphate from the ATP and we get ADP plus some energy. So remember, the source of the way that the molecules are storing the energy is in those chemical bonds, specifically those covalent bonds. Okay, so the Calvin cycle, that's the second step of photosynthesis. It takes place in the stroma, and it takes the products, the ATP and the NADPH, from the light reactions and uses them to make sugars, carbohydrates. It doesn't require light. It can happen in the light or the dark, so that's why we don't want to call it the dark reactions anymore. And It is an extremely complicated process. They're still studying all of the chemical reactions in the Calvin cycle. Just know that this cycle is 90% efficient in trapping the energy that's released from the breakdown of the chemical bonds and putting it into glucose molecules. So it's extremely efficient. It's pretty amazing. These reactions are going to happen with or without light. And we start out with that rubisco and carbon dioxide. So this is where our carbon dioxide comes into play. Okay, our carbon dioxide comes into play from the light reactions. We've got those hydrogens left over, those ATPs left over. Water, this is why we have water on our right side of our equation when we write the more correct equation for photosynthesis. And this ribulose bisphosphate, RUBP, here gets recycled. This is why it's a cycle. It gets sent back up here, and we get glucose. And then, of course, carbon dioxide is a waste gas given off. Don't forget to watch that animation there. And moving on. So, remember that we have the ATP and the NADPH as chemical bond energy, which gets converted into another chemical bond energy, just this time in our glucose molecules. Okay, so we go from chemical storage in these two molecules into glucose because our cells can't do anything with this NADPH. This ATP is not enough to fuel the cell. Basically, all of the energy here goes into the chemical bonds between the carbons, the hydrogens, and the oxygens here. And remember, we discussed in class, what happens to the glucose? What's the fate of that glucose? That glucose then goes into the mitochondria. And those are the cellular respiration reactions, which we're going to talk about in the next chapter. I don't think you really need to learn this step for our purposes here. Just know that we've got carbon dioxide coming in, and RUBP is where we started all off. And there are a series of chemical reactions. This gets even more complicated than what's shown here. But these molecules, this is unstable. It starts out as a 6-carbon molecule, and it immediately gets broken down into two 3-carbon molecules. From there, it gets switched again. The hydrogen comes off of the NADPH, gives it a little bit more energy, a little bit of ATP comes in, and the end result is a molecule of glucose being made. And this molecule of glucose then, see how it refers to going to cellular respiration in the mitochondria? But what if the cell has a whole bunch of glucose around and it doesn't need it right away? Well, it's going to store it. And it's going to store it as a long chain of glucose molecules, which is a starch. And remember, hopefully from your macromolecules unit, a starch molecule is a polysaccharide. Lots of single glucose molecules strung together. And it would be stored in the stem or in the root system. Okay, so here is an overview. More detail than you need. In summary, the Calvin cycle fixes, that means it traps or it holds on to the carbon dioxide, specifically the carbon, and regenerates, remakes the RUBP at the end of the cycle. And then the PGAL is what gets turned into glucose. So, which part of those processes occur in the light? Which part? Well, you'd be right if you said the light reactions. That's why we call them the light reactions. Which process can occur in the dark? Ooh, I made a mistake. Did you make the same mistake I did? Let's go back to that first question. Which parts of the process can occur in the light? Is it just the light reactions? And if you're thinking, well, no, you're right. It's the light reactions and the Calvin cycle. Okay. Which parts of the process can occur in the dark? And that would be only the Calvin cycle. Only the Calvin cycle, because it's light independent. How is the structure of the chloroplast related to its function? I know we've talked about this in class. Remember all those inner folded thylakoid membranes? Well, it gives us lots of surface area with lots of chloroplasts, or chlorophyll embedded in that surface area of the membrane, and so we get a lot of absorption possible. Here's a great link to a PBS website and some videos that you can watch. Let's look here quickly at the structure of a leaf. You will see some of this on your quest tomorrow. Remember, we've got this upper cuticle layer and lower cuticle layer, which minimizes the water loss. Okay, it's going to be a waxy cuticle. Then we have an upper epidermis and a lower epidermis. This is just basically like the skin of the leaf, just like we have an epidermis. It's this layer, the palisade layer. A mesophyll means middle of the leaf. And so you see how these cells are all lined up in a columnar, like columns. And this is to maximize the wavelengths of light passing through so that it maximizes sunlight absorption. Down here in this spongy layer, it's called spongy because there's all these open air pockets, and this is because we need to have plenty of space to store carbon dioxide and oxygen. On the lower epidermis, we have, remember, the stoma, the openings that allow for gases to come in and out, and then the guard cells, which can open and close the stoma. And there's a beautiful picture of them and another one there. And there is some stuff. Now remember, this lecture, if you don't want to listen to me or if you just want to click through the slides at your own pace, is also on School Loop on the calendar. Okay, so here's a nice review. We have ultimately the energy from the sun getting trapped as chemical bond energy. And I'm frozen again. Here we go. And I think I am just about at the end of this. So I'm hoping that this helps. There is another really short PowerPoint that I put up with just a kind of an overview, some review material for you. Don't forget, there's all those great Bozeman science videos and crash course videos that you can watch. So please like this video, give me some comments, and if you have any questions, email me tonight. I'll try to get back to you. Talk to you soon. See you in class tomorrow. Bye.